Hello everyone, my name is Mariely Vega. I am a PhD student from North Carolina State University, and today, as my title says, I will be talking to you about macroinvertebrate functional feeding group response to drought and hurricane disturbances in the tropical mountain streams of Puerto Rico. So to start off, tropical mountain streams can be very geomorphologically and hydrologically variable, and that natural variability is one of the main factors driving stream community dynamics. Individual taxa in these assemblages can be classified into functional feeding groups, or FFGs, which considers the mechanisms behind their feeding strategies and how they consume the available resources that they have in their habitat. So the classification for functional feeding groups include the collector-gatherers, the scrapers, the predators, the filterers, and the piercers. In the Caribbean region, natural phenomena such as droughts and hurricanes notably alter those resources which are available for freshwater organisms to consume. So why is this important? In 2015, Puerto Rico experienced one of the most intense droughts in the previous 2 decades. So over 60% of the island fell under moderate to extreme drought classifications, and there was long-lasting water rationing that took place across the island given the substantial decrease in water level to our streams and our reservoirs. Another important event occurred in 2017 when two major category hurricanes, Irma and Maria, impacted the island, causing an immediate increase in discharge, which was followed by a widespread defoliation of the riparian forest. My objective in the study was to evaluate the effects of the 2015 drought and 2017 hurricanes on freshwater ecosystems by assessing changes in macroinvertebrate functional feeding group composition and abundances. My study was carried out in the island of Puerto Rico. Puerto Rico is the smallest island of the Greater Antilles, located in the Caribbean, and my study streams are in the northeastern part of the island, in the El Yunque National Forest, or Luquilla Experimental Forest as it is also called. Sampling occurred in two low-order and convergent streams called Quebrada Prieta A and Quebrada Prieta B. For the methods, macroinvertebrate collection occurred approximately quarterly from 2014 to 2015, then monthly after there was a change in protocol from 2016 to 2018. In this study, I looked exclusively at the pools, where collection is carried out by placing a quarter in the pool, as you can see on the image to the left. Then we transfer the material from the bentos into a bucket, where we later sieve the material and preserve the organisms to identify them later. After the organisms have been preserved, we transported them to the lab, where we identify them to the lowest possible taxonomic level, which would be usually the genus. Then we looked in the literature for assistance in classifying the organisms into their respective functional feeding groups. What followed was determining discrete temporal periods around those disturbances to evaluate if any changes had occurred from one group to the next in terms of the functional feeding group abundance. To assign these groups, we use a combination of long-term and short-term precipitation data, such as the one depicted on the figure to the left, and the resulting groups consisted of two drought groups, a pre-drought group and another group during the peak of the drought. For the hurricanes, there was a total of three groups. So we have before the hurricanes, post-hurricane, and short-term post-hurricane. For the drought, we looked at the individual samples for a total of 4 months, and during the hurricanes, we looked at the average abundances of functional feeding groups for a total of 9 months. So after we defined the groups, we ran non-metric multidimensional scaling and MDS analysis to observe changes in assemblages following the disturbances. We also ran a permanova to determine if there were significant differences among the groups we had delimited, and additionally, we ran simple analysis to determine which taxa were responsible for causing the greatest dissimilarity among those groups. Okay, so now let's move on to the results. This is the result of the MMDS during the drought. Something important to note about this figure is that points that are closer to each other have assemblages that are more similar than points that are farther apart. The blue points represent the samples collected during the pre-drought period, while the red points represent the samples from the drought group. Visually, we don't see that the points separate into distinct groups based on the periods. However, we do see that some pools did change during the drought, but the variability of the functional feeding group abundance was very large. So now if we look below the figure, you'll see that after running a permanova to assess the differences among the periods that we delimited, no significant differences were found between the groups. Notably, when we explored the data further, by running a simple analysis, the collector-gatherers, like the one in the image below, were responsible for most of the differences between the assemblages. So what this figure shows is the abundance of the individual samples per group, which to reiterate were 4 samples for each sampling month. It shows the high variability within the groups and among the groups, but also that there were peaks in the collector-gatherer abundances that occurred during June and July of 2015 during the peak of the drought. And even though these occurrences are not uncommon, as you can see from the point on November of 2014, the maximum values for FFGs were up to 5 times higher than the previous peak, particularly when we looked at the collector-gatherer abundances. Okay, so now let's move on to the results from the hurricanes. In this case, you'll see that each data point within a group consists of the monthly average, and each group is composed of 3 sampling months. We started by looking at the NMDS results, where you can see the before hurricane group in blue, the post-hurricane group in red, and then the short-term post-hurricane group in green. When we look at QPA, we see that 3 groups can be visually distinct, and they have this very interesting, almost circular pattern, as you can see here. And this pattern goes from before the hurricane, post-hurricane, and then short-term hurricane, in which we can almost predict where the assemblage would be headed given enough time. With these observations, we could perhaps hypothesize that FFG assemblage will resemble the before hurricane group if the system is allowed to recover undisturbed. When we look at the Permanova results to compare among periods, we see that the groups are significantly different. And even though when you look at the function of feeding group abundance across time, you see that there is a lot of variation, it is very notable that there is an overall decrease in the abundance of the groups after the hurricanes. In contrast, when we look at the NMDS for QPB, there are no distinct groups that arise after the events, but note that the spread of the data seems to be larger for the post-disturbance groups when they're compared to that group before the hurricanes. When we look at the Permanova results, we see that no significant differences were found among the different groups. Additionally, when we look at the FFG abundance figure on the right, we see that the overall FFG abundance decreases sharply after the hurricane, and then it starts increasing until it reaches a peak in the collector-gatherer abundances up to 4 times before decreasing sharply again. If you look at the NMDS analysis for the streams together, so QPA and QPB together, you will see how the patterns now seem to be more closer to that of looking at QPB by itself, in which groups are not entirely distinct, variability of the data tends to increase after the hurricanes, and no significant differences were found between the groups. If you notice the FFG abundance figure, the patterns after the hurricane resemble again that of QPB trends more closely than the QPA trend. The most notable response to the hurricane is a decrease in FFG abundance, which is followed by an increase, particularly in the collector-gatherers, and then a sharp decline during the last month of the short-term post-hurricane group. The simple analysis for the hurricane groups confirms that the collector-gatherers and in a single case the scrapers are the tags are responsible for causing the greatest dissimilarity between the assemblages of the groups. So what does this all mean? We saw that the drought caused peaks to the collector-gatherer abundance, but that the patterns were not consistent among the pools. So something that could potentially be taking place is that during the peak of the drought, the largest pools in QPB might have been like the smaller pools during non-drought years, and this might help explain that lack of significant differences. We propose that the initial increase in water level could be responsible for the initial decrease in the function of feeding group abundance, and then changing basal resources during the following post-hurricane months might be the responsible for the notable increase in the collector-gatherers that followed. We did find significant differences between the groups due to the hurricanes in QPA, where the response of the assemblages was clear. We suggest that due to the hurricane, that initial increase in water level could be responsible for the initial decrease in function of feeding group abundance, but then due to the cyclonic winds, a change in the basal resources during the following post-hurricane months might be the responsible for that notable increase in collector-gatherers that followed. In QPB, the response was more variable and not consistent between the pools. We can see that the sites responded differently. This is evidenced by looking at the streams together, because we see that more variable pattern of response, driving the point home that response was site-dependent and could be driven by variations in geomorphology from pool to pool. Additionally, interpretation from looking at 2 seemingly comparable streams should be conducted carefully and considering local variations in channel structure. The next steps consist of evaluating response to natural phenomena, integrating hydrology and geomorphology in the study designs. To conclude, patterns suggest that individual pool geomorphology plays an important role in determining macroinvertebrate response to disturbance. A comprehensive understanding of how stream ecosystems respond to disturbances is critical to understand their vulnerability to predicted changes in environmental conditions due to climate change. Lastly, I would like to thank the incredible and immense group of people from a variety of institutions that contributed to making this study possible. And thank you for watching my video. I look forward to your questions. Bye!